Hi all. I will be talking about iron deficiency anemia. In order to understand iron deficiency anemia, you first have to understand what is hemoglobin. Hemoglobin is a protein found in the red blood cells. Its main function is to transport oxygen to the tissues and carbon dioxide from tissues to the lungs. At high oxygen pressure, meaning when a person is exposed to oxygen, hemoglobin then has a high affinity for oxygen. And at low oxygen pressure, hemoglobin has a low affinity for oxygen and therefore releasing the oxygen to tissues. So hemoglobin is basically made up of two parts from its name, which is heme as well as globin. Heme is made up of both iron and porphyrin ring, and globin, on the other hand, is made up from 2 alpha chains and 2 beta chains. This is in a normal adult blood. Iron deficiency anemia falls under a classification of anemia known as microcytic hypochromic anemia. And it is the common cause of anemia in worldwide, and most common cause of microcytic hypochromic anemia that I was just saying. So all red cell indices, your mean cell volume, your mean cell hemoglobin, as well as your mean cell hemoglobin concentration is decreased in iron deficiency anemia. Your blood smear will show microcytic as well as hypochromic cells due to the hemoglobin defect in iron deficiency anemia. So these will usually present as pencil cells as well. So these are basically flat cells with low osmotic fragility in most cases. So the differential diagnosis of iron deficiency anemia include other microcytic hypochromic anemias such as your thalassemia, anemia of chronic disease, or infection. The main cause of microcytic hypochromic anemias is basically your lack of iron, known as obviously the iron deficiency, your decreased release of iron from the macrophages or storage, which is seen in your anemia of chronic diseases, and the failure of protoporphyrin synthesis, which presents as an anemia known as sideroblastic anemia, as well as failure of globulin synthesis, where we see our alpha and beta thalassemia. The causes of iron deficiency anemia include, but is not limited to, chronic bleeding, increased demand in the case, for example, of pregnancy, decreased absorption, decreased diet intake, and increased loss in the case of many things, pregnancy, blood loss. So a variety of things can lead to iron deficiency anemia. The symptoms of iron deficiency anemia firstly include the symptoms of anemia, which is your shortness of breath, your skin pallor, most noticeably in the lips as well as in the inner part of the eye, dizziness, loss of breath, unusual malaise. Then you have symptoms of iron deficiency anemia itself, which include glossitis. So it's usually a red beefy tongue due to lack of iron. You have your nails become brittle and spoon-shaped, most noticeably. Heart palpitations, as well as strange cravings such as, or known as rather, pica, where you crave to eat certain things like dirt, clay, ice, chalk, paper, cold hands and feet, cold skin. This is usually because, remember, the hemoglobin brings color as well as oxygen to your hands and feet, and when hemoglobin is deficient, then this is usually the result in most cases. Hence, these are the symptoms of iron deficiency anemia. The pathogenesis of iron deficiency is actually quite simple and occurs in 3 stages. Initially, your iron stores are depleted, hence iron deficiency. So your iron stores are deficient. Secondly, your serum iron and as well as your transferrin saturation are decreased, and your total iron binding capacity is increased because you have a deficiency of iron, right? Both your serum iron and your transferrin saturation, transferrin, which is responsible for carrying iron, will be decreased. However, your body has a high affinity, has a high want to bind iron. Hence, your total iron binding capacity will be increased. Your iron deficiency anemia, being the last stage, will then manifest when HB is decreased. So when your hemoglobin is decreased, that's when iron deficiency anemia manifests. This is usually due to the fact that your bone marrow has depleted. Your bone marrow iron stores have been completely depleted. So when you go to the doctor, take your blood and send tests just to confirm that you are iron deficient, but not only iron deficient, but you are suffering from iron deficiency anemia. So your full blood count, also known as complete blood count, will be your hemoglobin will be low, your hematocrit will be decreased, as well as your red cell indices. These include your mean cell volume, mean cell hemoglobin, and mean cell hemoglobin concentration will all be decreased. Your white blood cell count will in most cases be normal. However, your platelet count, your thrombocytes, will be high or normal, especially when bleeding is involved and chronic in some cases. And you generally present with anisocytosis, which by definition means a variation in red blood cell size. And furthermore, your peripheral blood smear. Remember that along with your test, in most cases, your peripheral blood smear will be made if your hemoglobin is low in most laboratories. So your peripheral blood smear will show microcytic and hypochromic cells, red blood cells, meaning that your red blood cells are pale. Target cells will be present. You will see or observe poikilocytes, and in most cases, it also depends on how far along the anemia is, or patients who have been diagnosed and are being treated. In the case of treatment, a dimorphic picture can be observed. Basically, a dimorphic picture is microcytes and ovalocytes that occur as a result to both folate or B12 deficiency, and in some cases iron deficiency during treatment stage. So that's what you see. You will also see anisocytosis on the peripheral blood smear, and your most common feature of any anemia, especially iron deficiency anemia, is your pencil cells that I had earlier mentioned. They are the clue. They are the key to iron deficiency anemia. Obviously, you need to ensure that the pencil cells are indeed of microcytic hypochromic anemia cause and not of other anemic cause in the case of hemolytic anemias. Special tests include your hemoglobin electrophoresis, which in most cases would be normal. Just osmotic and fragility is not really much done test in iron deficiency anemia, but it is done in some cases, of which will be low because poikilocytes or your pencil cells are flat. And your most valuable diagnostic test are your iron studies, which is conducted in chemistry. For iron deficiency anemia, the results of your iron studies are as follows. Your serum iron is low. Your total iron binding capacity will be high. Your serum ferritin will be low. Bone marrow iron stores will be absent. remember that in order for you to be diagnosed as iron deficient anemic, your bone marrow iron stores have to be completely deficient, okay? And your erythroblast iron will be absent as well. Your hemoglobin electrophoresis, however, will be normal. So that is basically it. That's basically everything on iron deficiency anemia with regards to what it is, the pathogenesis, and in the lab, what exactly is the doctor looking for in order to diagnose you as iron deficient anemia.